faithfully sharing value and interpreting great books from around the world. Hi, this is Sally here with the Book Talk. Today we're diving into the book called Survival of the Sickest. In today's world, health is definitely one of the top concerns for people. Many folks spend tons of time and money each year on health and wellness, hoping to avoid diseases. But even now, humans can't seem to shake off diseases. Is it cause diseases are too strong, or cause medical tech isn't good enough? Nope, neither one. Actually, often diseases are an active choice by human genes, though some, like diabetes or high cholesterol, just bring trouble now. But in human evolution, these diseases helped us fight environmental changes and extend our lifespan, like good buddies. The relationship between humans and disease is way more complex than we think. The author Sharon Malelm holds dual doctorates from the University of Toronto in Canada and Mount Sinai School of Medicine in New York, USA. A neurogenetics expert, he's also a science writer with books like The Sexless Drive, Reboot, and DNA. In Survival of the Sickest, the author dives deep into human evolution, offering a fresh view on life, our bodies, and how we understand disease. I've broken down the whole book into 3 parts to explain it to you. Part 1, I'll cover some common diseases caused by genes. Part 2 explains from an epigenetics perspective why genes trigger these diseases. Part 3, I'll share what humans can do about these genes. Let's start with diseases caused by genes. First up is hemochromatosis, a metabolic disorder from too much iron buildup in the body. Disease? Can iron actually cure illnesses? You see, people view iron as a key helper for health. From childhood, we've all had iron-rich foods like pork liver or spinach or red peanut skins. People are so into iron because it makes sense scientifically. Our bodies don't make iron, so we got to get it all from outside, and it's vital for metabolism. Without iron, oxygen from the lungs can't be moved around the body. Iron is also a key component of many enzymes that convert sugar into energy, keeping our bodies running daily. If iron levels are insufficient, people may develop a range of illnesses. Take anemia, for example. People with iron deficiency anemia often appear pale, experience shortness of breath and fatigue, have weakened immunity, and in severe cases, it can be life-threatening. But is more iron always better? Absolutely not. This is a common misconception people often fall into in daily life. Though iron is essential for our bodies, it's definitely not the more the better. In fact, too much iron supplementation can actually make people more susceptible to illness. For instance, in the 1970s, New Zealand doctors believed the indigenous Maori population had subpar living standards, especially noting significant iron deficiency in infants' diets. So they launched mass iron supplementation injections to improve Maori health. So, surprisingly, fatal diseases among iron-injected Maori infants surged dramatically. Rates of sepsis and meningitis were 7 times higher than in other infants. Scientists later discovered that while these pathogens easily infect infants, their reproduction rate was naturally slow. After iron supplementation, these pathogens' reproduction rates skyrocketed. Similarly, Dr. John Murray observed in Somali refugee camps that while iron supplements cured anemia in malnourished refugees, infection rates for malaria and tuberculosis also surged dramatically. Why does this happen? Turns out, not just our bodies, bacteria, fungi, and viruses also need iron to reproduce. To these pathogens, our iron-rich blood and tissues are like gold mines. Excessive iron intake is like serving them a daily, all-you-can-eat buffet, allowing pathogens to multiply wildly and drastically increasing infection risks. Fortunately, for most people, moderate iron intake poses no harm. But for those with hemochromatosis, iron supplementation is nothing short of a disaster. Hemochromatosis is a genetic disorder especially prevalent among people of Western European descent. Normally, our bodies self-regulate When iron reaches certain levels, we automatically reduce absorption and increase metabolism to maintain balance. But for people with hemochromatosis, their bodies constantly feel iron deficient, so they keep absorbing it from the gastrointestinal tract, eventually depositing excessive iron. This excess iron damages joints and organs, ultimately causing death. You've probably heard evolution's core principle is survival of the fittest, meaning if a genetic trait makes humans stronger, it gets passed to the next generation. Conversely, traits that weaken us are unlikely to be inherited. Over time, natural selection reads out troublesome genes, keeping those that strengthen us. So here's the puzzle. Why does a killer like hemochromatosis persist stubbornly in our genes? There's only one reason: it benefits us. While hemochromatosis hoards iron, its distribution is uneven. Excess in organs like liver and pancreas, but critically low in macrophages. These immune cops patrol our bodies, surrounding and devouring pathogens to destroy them in lymph nodes. Normally, iron-rich macrophages risk supercharging captured pathogens, boosting their reproduction and infection power. But hemochromatosis patients' iron-poor macrophages avoid this danger. Hearing this, you might ask, doesn't hemochromatosis actually help immunity? Then why call it a killer? Because while it won't kill you immediately, it slowly causes fibrosis and degeneration in heart, liver, pancreas. Simply put, these vital organs deteriorate irreversibly with current medical treatments. Compared to these devastating outcomes, that minor immunity boost is nearly meaningless, unless right now you're facing a terrifying infectious disease that could kill you at any moment. Infectious disease. You might have heard of the plague. It's also called the Black Death. It's the most famous and deadliest infectious disease in European history. In the 1347 plague outbreak, nearly 30 million died across Europe, almost half its population then. But hemochromatosis carriers with iron-poor macrophages showed strong plague resistance, surviving and passing the gene to descendants. Over the next 300 years, as plague resurged repeatedly, carriers multiplied, embedding this gene firmly in Europe's gene pool. If I asked why you'd take a drug that kills you in 40 years, you'd say, because at least it lets me see tomorrow's sun. Similarly, why keep a gene that might give you hemochromatosis decades later? Because it protects you now from certain death, sparing you an early grave. So in human evolution, our relationship with iron can't be judged as simply good or bad. Moving from hemochromatosis, let's discuss another widespread disease today: diabetes, one of the world's most common chronic illnesses. WHO data shows approximately 170 million people have diabetes globally. But what actually causes it? We still don't fully know, generally blamed on genetics, infections, or poor diets. Medically, it's split into type 1 and type 2, mainly differing in insulin levels. Type 1 involves plummeting insulin production, causing severe shortages. Type 2 usually produces insulin normally, but the body rejects it, blocking its function. Studying diabetes distribution, scientists found something fascinating. Type 1 is most common in Northern Europe. Finland leads globally, Sweden second, Norway and UK tied third. With rates dropping further south, diabetes is rare among Africans and Hispanics. Just like with hemochromatosis, we've learned that when certain traits become prominent in specific environments, they usually benefit humans. So, could higher blood sugar in cold regions also be beneficial? Before explaining this, let's consider winemaking as an example. You might know ice wine, made from frost-exposed grapes. Freezing reduces their juice but concentrates sugar, creating exceptionally sweet wine. This is actually the grape's self-protection mechanism against freezing temperatures. Similarly, humans develop comparable defenses in cold environments. For instance, in cold seasons, you might pee more even without drinking extra water. Like grapes reducing internal moisture, this is our body's self-preservation tactic. Another example: blood sugar rises when it's cold. Why does this happen? Obviously, we can't freeze humans to test this. So, scientists experimented with Alaskan wood frogs, small creatures needing hibernation to survive extreme cold. Unlike other hibernators, wood frogs don't just sleep deeply, they completely freeze solid. Frozen wood frogs show no heartbeat, breathing, or vital signs, essentially becoming ice blocks. Yet, come spring, they thaw and hop away perfectly fine. Blood sugar plays a key role here. Research shows when hibernation starts, their livers flood the bloodstream with sugar, spiking blood glucose hundreds of times higher. This sugar lowers their blood's freezing point, and high concentrations create smaller, rounder ice crystals, preventing sharp crystals from rupturing capillaries and minimizing damage. Um, so could Northern Europeans' higher blood sugar similarly protect against cold? Exactly. Archaeological findings reveal around 13,000 years ago, Earth experienced a sudden cold snap called the Younger Dryas period. Initially, scientists thought this dramatic climate shift lasted thousands of years. But in 1989, new research suggested it might have lasted just 3 short years. Imagine global temperatures plummeting 30 degrees in just 3 years. How terrifying. Mammoths, saber-toothed tigers, cougars, all wiped out during this period. Early humans suffered too, like the Clovis people who went extinct from the cold. Yet Northern Europeans survived. Picture this small group in the freezing cold, slower insulin release, higher blood sugar than normal. High blood sugar lowered their blood's freezing point, reducing cold damage. This gave them a survival edge in the extreme cold. That edge allowed survival and passing cold-resistant genes to descendants. So high blood sugar actually helped Northern Europeans' ancestors survive sudden freezing. But modern diabetics rarely face sustained cold. Their excess blood sugar becomes harmful instead of helpful. Still, these troublesome genes are ancestral battle scars from humanity's fight against nature millennia ago. Diabetes is just these genes announcing their presence. Since we're discussing genetic trade-offs, here's another example showing how identical genes behave differently across environments. Let's explore vitamin D production. It's deeply linked to cholesterol in our bodies. Vitamin D, crucial for metabolism, plays an irreplaceable role in childhood bone development and adult skeletal health. You've probably heard elders say, kids need sunlight to absorb calcium and prevent rickets. So why does sunlight help us get vitamin D? Turns out our bodies have an amazing system converting cholesterol to vitamin D using UV rays from sunlight. More sun exposure converts more cholesterol into vitamin D. Less sun means less cholesterol gets converted. So when your checkup shows higher cholesterol, don't panic immediately. First, check the weather. Weak winter UV rays likely leave more cholesterol unconverted than in summer. Now, consider Russia, Canada, Norway, Finland. They get far less sunlight than equatorial regions. Does that prevent sufficient vitamin D production? And are their bones more vulnerable? Actually, no need to worry. Humans have the ApoE4 gene for this, produces apolipoprotein E4 that rapidly boosts blood cholesterol. With ample cholesterol, they maximize UV to vitamin D conversion, compensating for low sunlight. But like all compensatory traits, this genetic expression comes at a cost. Low sunlight areas have more people with high cholesterol. High cholesterol causes plaque buildup in atherosclerosis, drastically increasing cardiovascular risks. All these diseases share two things. First, they're tied to environmental impacts on our bodies. Second, they manifest through gene expression. So, how do environmental cues guide or even control our genes to create these fascinating changes? That's what epigenetics, a genetics field, specifically studies. In classical genetics, all biological traits are determined solely by genes. Trait changes must mean genetic changes. The primary change mechanism is genetic mutation. Simply put, you should inherit your parents' genes unchanged. But during inheritance, random events alter some genes, making them different from your parents, Totally unpredictable. You could turn into Spider-Man or end up a frail patient. Mutations are passive, but Darwin's survival of the fittest means beneficial ones get passed on, while harmful ones die out. Mutations can come from internal causes, like radiation messing with DNA replication, chemical interference, or external forces, viruses inserting genes into host DNA. Regardless of cause, whether mutations strengthen us is pure luck. If temperatures plummeted tomorrow, would mutations create cold-resistant humans? Only heaven knows. Relying on this genetic lottery makes species helpless against environmental shifts, doomed to extinction. But genetics isn't that simple. Geneticists now realize life has another system controlling genetic changes alongside mutations. Scientists call this epigenetics. Simply put, how offspring inherit and express new traits from parents without DNA mutations. Epigenetics is a very young field, conceptualized in the 1940s, but only in 2003 did major breakthroughs occur. At breakthrough, scientists experimented with mice carrying the agouti gene, affecting fur color and obesity. This gene gives mice yellow fur and makes them prone to obesity. When these mice bred, they always produced yellow, chubby babies. But when Duke University added prenatal vitamins to mothers' diets, they shockingly gave birth to thin, brown-furred pups, stunning geneticists worldwide. You might ask, is this a genetic mutation? Not at all. Scientists repeated with other yellow mice. After vitamin supplementation, they also produced thin brown mice. Genetic tests confirmed their agouti gene remained perfectly intact on chromosomes, meaning these genes weren't damaged, just silenced, like a switch was flipped. This silencing process is DNA methylation, simply methyl groups attaching to genes. Without altering DNA sequences, it changes how genes express, yielding different outcomes. Duke University's discovery massively impacted genetics, but overturned classical genetic theories. Diabetes is simply these genes announcing their presence. Since we're discussing genetic trade-offs, here's another example showing how identical genes behave differently across environments. Let's explore vitamin D production. It's deeply linked to cholesterol. Vitamin D, crucial for metabolism, plays an irreplaceable role in childhood bone development and adult skeletal health. You've probably heard elders say, kids need sunlight to absorb calcium and prevent rickets. So why does sunlight help us get vitamin D? Turns out our bodies convert cholesterol to vitamin D using UV rays from sunlight. More sun exposure converts more cholesterol into vitamin D. Less sun means less cholesterol conversion. So when your checkup shows higher cholesterol, don't panic immediately. First, check the weather. Weak winter UV rays likely leave more cholesterol unconverted than in summer. Now, consider Russia, Canada, Norway, Finland. They get far less sunlight than equatorial regions. Does that mean they can't produce enough vitamin D? And are their bones more prone to problems? Actually, no need to worry at all. Humans have the ApoE4 gene for this, produces apolipoprotein A4 that rapidly boosts blood cholesterol. With ample cholesterol, they maximize UV to vitamin D conversion, compensating for low sunlight. But like all compensatory traits, this genetic expression comes at a cost. Low sunlight areas have more people with high cholesterol. High cholesterol causes plaque buildup and atherosclerosis, drastically increasing cardiovascular risks. All these diseases share two things. First, they're tied to environmental impacts on our bodies. Second, they manifest through gene expression. So how do environmental cues guide or even control our genes to create these fascinating changes? That's what epigenetics, a genetics field, specifically studies. In classical genetics, all biological traits are determined solely by genes. Trait changes must mean genetic changes. A primary change mechanism is genetic mutation. Simply put, you should inherit your parents' genes unchanged. But during inheritance, random events alter some genes, making them different from your parents. Totally unpredictable. You could turn into Spider-Man or end up a frail patient. Mutations are passive, but Darwin's survival of the fittest means beneficial ones get passed on while harmful ones die out. So, mutations can come from internal causes like radiation, messing with DNA replication, chemical interference, or external forces, viruses inserting genes into host DNA. For instance, many viruses can integrate their genes into the host's cellular DNA. No matter the cause, whether humans get stronger is completely up to chance. Suppose temperatures plummet again tomorrow. Could anyone among us have a genetic mutation? And could that mutation create a group resistant to extreme cold? The answer probably only heaven knows. If survival hope relies entirely on God occasionally rolling dice, then such a race is undoubtedly waiting helplessly, lacking adaptability to the environment. Their only end is extinction. But genetics isn't as simple as you think. The more geneticists are realizing that Earth's life forms, including humans, while enjoying changes from genetic mutations, have another system controlling genetic changes. Scientists call this system epigenetics. Simply put, it's about how offspring inherit and express new traits from parents without genetic mutations. Epigenetics is a very young field. Its concept was first proposed in the 1940s, but not until 2003 did scientists achieve major breakthroughs in research. What exactly was the breakthrough? Here's what happened. Scientists used lab mice with a gene called the agouti gene. This gene gives mice unique light yellow fur and makes them prone to obesity. When these mice bred with each other, they always produced yellow offspring. Chubby baby mice. When Duke University Medical Center experimented with these mice, they added prenatal vitamins to the mother's diets for better care. Unexpectedly shocking genetics. The mothers gave birth to thin, brown-furred pups. You might ask, is this a genetic mutation? Not at all, scientists repeated with other yellow mice. After vitamin supplementation, they also produced thin, brown mice. Genetic tests on these brown mice showed their agouti gene remained perfectly intact on chromosomes. In other words, these genes weren't damaged, but now they're silenced, as if a switch was turned off. The silencing process is called DNA methylation. Simply put, it's a methyl chemical group attaching to genes. Without altering the DNA sequence itself, it changes how genes express, creating different outcomes. Duke University's discovery had a massive impact on genetics.